रिकॉर्डिंग अभी से क्यों करा चालू? अभी नहीं करी, दिखा रहा है मेरे को आवाज आई रिकॉर्डिंग इन प्रोग्रेस.
So I also welcome the participants who are joining this seminar and going to gain a lot of knowledge regarding the testing protocol and stability studies, which is comparatively a newer topic, especially for the Ayurveda, Siddha and Unani drugs. So on behalf of my experience and various observations, I would like to put forward few of the points here in front of panelists and government authorities also. Dr. Kasmi is here. So I hope that these points will be taken care in future. So especially testing protocol is a challenge, especially for ASU drugs, because most of the ASU drugs are multi-components drug, and these components or ingredients are going to have a lot of interference in the testing. So the person who are sitting here as a panelist and having the testing experience and qualification, they will admit with my comments that a lot of interference is there in the testing, and in many cases we need to develop the validated method for the products and different dosage forms. So this is one of the biggest challenge, and we can see that in case of Ayurveda, many a times we are not getting the results which are going to be reproducible, and a lot of variation is also there. So a lot of work on standardization of testing protocol is required. So the panelists who are sitting here from different test houses, they can suggest that which kind of testing protocol should be preferred for which kind of dosage form. So in day-to-day working, I have observed that the same testing protocol is not going to work for each and every Ayurvedic products. So different protocols are required. And regarding the stability, this is a very new area for the ASU drug because earlier the requirement of stability was not there. And in Ayurvedic pharmacopoeia, we can see that the part 2 different volumes have been set up there, and these volumes are having the specification along with the ranges of different parameters. So I have few of the observation for these parameters and their ranges also. So we can see that for different parameters like SG, pH, the quantification of markers, a very narrow range, and somewhere we can see that a firm quantity is also written. So you can see that a pH range like 3.09 to 3.10. This kind of range, are the total tannin content 2.01. This kind of parameters with different observations are there in the API part 2 different volumes. So these are required to be taken care. And one thing that is more important here, that during the stability study of any of the product, the packaging and the storage condition are the most important points for any kind of stability. Before putting any stability observation, the packaging details must be there. So I would request that in future, if API is going to have any specification based on the stability, though I doubt that these specifications are based on the stability. So if we are going to develop and put any specification in the API, it must have the details of the packaging and container closure system also. Few other observations I would like to narrate here, that in case of ASU stability, different conditions are mentioned there in the API part 2, and I am not able to remember the exact volume where the detailed instruction of the stability evaluation have been narrated. So it is written there that the variation in the physico-chemical parameters should not be more than 25%. So suppose if we are going to evaluate the LOD, and that LOD is 4% in case of a powder or in a herbal tablet. And if its LOD during the stability evaluation goes beyond 5, certainly it is going to be fail in the stability studies. Accordingly, the range given for the acid value, peroxide value, and SG and pH is very narrow. So these kind of things are required to be taken care. And during the development of specification, it is very much required to take care of the details of the composition also. Suppose we are going to develop a specification for Sitopaladi Churna. And for a minute, just agree with me that Sitopaladi Churna by composition has 25% of Bansalochan in its composition. So Bansalochan is a fumed silica or silicon dioxide, you can say. And its total quantity is going to be converted in the AS. So AS value of that product will be 25% of the FG quantity, which will be taken for the evaluation. By the virtue of Bansalochan only. And if we are going to design a specification where its total AS content is going to be lesser than 25%, then it is not going to be the effective specification. It will be a faulty specification. So these kind of important parameters are required to be taken care during the development of stability, during the evaluation of stability, because the Ayurvedic parameters by nature, by composition, have different qualities in comparison to the allopathic compounds. We are having different components. By nature, they are having the tendency to absorb the moisture. Ayurvedic tablets gain moisture. Their thickness changes. You can say their diameter changes by nature. In due course of time, if you are going to evaluate, the thickness and diameter of the Ayurvedic tablets in any kind of container closure system will vary during the stability study. Accordingly, the acid value or peroxide value of different raw material oils like sesame oil and coconut oil and various other oils which we are using in the Ayurvedic pharmaceutics and different finished goods is going to have a lot of variation during the stability studies. So these are very important points, and on the basis of my experience, I wanted to highlight these issues in front of these learned panelists so that a very thorough discussion on these important points can be made here on this platform. So these are important points, and I once again welcome you all the panelists, and I hope that a very healthy discussion is going to be done here, and a lot of things will be sorted out, and a firm guideline and the things which we are going to discuss here in front of different government authorities will be taken care for future also. So I request the analytical panelists also to put forward their experience and observation, especially for the Ayush drug, in front of all the panelists and the attendees, so that they can also have the practical problems which you people are facing day to day in your observation and analysis. So with these kind of remarks, I thank you again and welcome for the next round of discussions. Thank you, Ravi.
Thank you so much, sir. Thank you so much for sharing your observations with us. We have seen the continuous attention of the Ayush drugs post-COVID. So it is the responsibility of our manufacturers also to provide the drugs with adequate stability for the long-term storage and the safety for the consumption by the patients. So thank you for sharing your insights on the stability protocols and the studies and your observations. Now I'd like to request our guest of honor, Dr. Pankaj Goyal. He is the Deputy Director at National Accreditation Board for Testing and Calibration Laboratories, NAPL, to share his remarks. We'll hear our guest of honor and chief guest for some more time with their key observations and the processes to learn from the government perspective also, and what are the uses we can get from the ministry. Over to you, sir.
Thank you, Dr. Ravi. I hope I am audible to all. Is my voice clear?
Yes,
Hello?
sir. Yes, sir, you are audible.
Okay. Very well respected chief guest of the event, Shri Kasmiji, Advisor, Ministry of Ayush, Padma Bhushan Ved Trigunaji, Patron of MM, Dr. Rajiv Raiji, Chair Ayush Committee, Dr. Joshi, Co-Chair Ayush Committee, Dr. Gunawat Ma'am from Max Lab, Sinha Saab from Multani Pharmaceutical, Agrawalji from the Hamdard Lab, and all eminent personalities present in this webinar. First of all, I would like to extend a warm welcome to all on behalf of NABL, National Accreditation Board for Testing and Calibration Laboratories. Like in the opening remarks by Dr. Rajiv Raiji regarding the concern of today's seminar, that is the testing protocols and stability studies of ASU drugs, and of course certain issues which we are facing in day-to-day, testing or the stability studies of the ASU drugs. First, I would like to give a slight glimpse of this good manufacturing practices which is required in any kind of the pharmaceutical industry, and which is to be maintained by the manufacturer as per the requirement of the Schedule M of the Drug and Cosmetic Act 1945. Well, GMP ensures that the products are consistently produced and controlled to the quality standards appropriate to their intended use, and GMP is aimed primarily at diminishing the risk inherent in any pharmaceutical productions. Well, here comes the importance of the stability testing as well. Like Dr. Rajiv Rai has told that earlier there was not any requirement of the stability studies, but now it is also very much required in ASU products also. The importance of this stability testing of pharmaceutical product provides the evidence of how the quality of an active pharmaceutical ingredient ABI or the finished pharmaceutical products FPP varies with time under the influence of a variety of environmental factors such as temperature, humidity, and the light. The stability testing program also includes the study of the product related factors that influences the quality of the product. For example, if we talk about the interaction of the API with the excipients, container closure systems, and the packaging material, then of course the stability plays an important role. As a result of the stability testing, a retest period for the API or the shelf life of the FPP can be established and storage condition can be recommended, so that on that basis, the how the ASU products can be manufactured, can be stored, or can be exported to different houses can be maintained. So it is very much important in terms of the requirement of the current scenario of the ASU portfolio, as like Dr. Ravi also said that post-COVID, there was a lot of improvement in ASU drugs. And there was also a lot of scenario being developed for the use of the Ayurvedic drug instead of the allopathic drugs, which is of having certain kind of the adverse effect. Well, having, from the background of the NABL, National Accreditation Board, so quality plays an important role in stability testing as well as the other testing protocols. And quality is never an accident. It is always the result of an intelligence effort. And effort always, this kind of the intelligent effort come from the standardization. And as all of you know that NABL works on the standard that is the ISO IC 17025, that is the testing for giving the accreditation to the testing and calibration laboratories. Before proceeding that, I would also like to, give the glimpse on general guidelines for the drug development of the Ayurvedic formulations that has been given by the CCRS, Ministry of Ayush. Of course, Kasmi Saab is there, so he will highlight this particular portion. So here, I would like to take one important point that Section 11, Part 2 said that a manufacturing unit shall have a quality control section as explained under Section 35.2. Alternatively, these quality controls provisions will be met by the getting testing, etc. from a recognized laboratory for ASU drugs, that is Ayurveda, Siddha, and Unani drugs, under the Rule 160A of the Drug and Cosmetic Act. And if we talk about the Rule 160A. So very recently, during July 2021, I think the Gazette of India notification comes with the amended Rule 160A that will says that the institutions for carrying out the test on ASU drugs and raw materials used in their manufacture on behalf of licensee for manufacture for sale of ASU drugs. So all such institutions which have facilities are required as required for the quality control section and let down under the Schedule T of the Drug and Cosmetic Act, and as well as should be accredited by the NABL, National Accreditation Board for Testing and Calibration Laboratories, for the category of chemical and biological testing for four major things, that is identity, purity, quality, and strength of ASU drugs and raw materials, of course. So it should be notified as the approved drug testing laboratories and for the purpose of this rule by the central government. So here comes the NABL is how it is required in all the kind of the testing laboratories which are testing the chemical or biological characteristics of any kind of the ASU drugs. Well, if we'll talk about, I'll say about the NABL, that NABL is the National Accreditation Board for Testing and Calibration Laboratories, and we give the authenticity of the test results on the basis that we are giving the test witness during the assessment of the laboratories, during which the competency of the personnel is judged, metrological traceability and the calibration of equipment is verified. We also ensure the validity of the results by the ILC and PT, that is the proficiency testing, and the quality management system. So all this done in accordance with the ISO IC 17025, which is under 2017 version of the standard. Like in the opening that one thing was raised about the validation and the verification of the method. So the standard ISO IC 17025-2017 clause 7.2.1 and 7.2.2 clearly says about the selection and verification of the methods as well as the validation of the methods respectively. So whenever any kind of the ASU testing laboratories are getting accredited in accordance with the ISO IC 17025-2017, they have to select an appropriate method for the testing of such kind of the ASU drugs. If the standard test method is not available, then they have to formulate the kind of the SOPs for the testing of that particular method, and they have to validate that method. Only after they will get the accreditation, and once they will be accredited, then these results are accepted everywhere as per the ILAC, APAC, MRA of NABL. So accreditation in a single line, if I'll say that it is the independent evaluation, that is a third-party attestation related to the conformity assessment body conveying the formal demonstration of its competency to carry out the specific conformity assessment task. So basically it is the three parts. One is the independent evaluation. That means NABL directly have the complete independent role for any kind of the laboratories which are doing the testing for any kind of the test parameter. So the accreditation will be given on the basis of the formal demonstration of the competency by the laboratory. If the laboratory is not competent, the accreditation will not be given for that particular test parameters. Accreditation ultimately gives the formal recognition of the competence to the laboratory, and it also ensures the sound quality management system of the laboratory. That will ultimately enhance the customer confidence in the satisfaction, and also reduces the technical barrier to the trade, and it gives the global acceptance to the report, which will increase potential business and market advantages as well, and give the laboratory the international recognition and also improves the staff motivation. So whatever the test reports issued by the NABL accredited laboratories will be acceptable amongst the economies which are signatories to the ILAC MRA. Like NABL is also having the ILAC MRA since 2000. And we are also, operate under the under the requirement of the ISO IC 17011, that is the conformity assessment. General requirements for the accreditation bodies, accrediting conformity assessment bodies. We are having the test, five kind of the accreditation services providing, that is the testing services and the calibration services as per ISO IC 17025, medical testing laboratories accreditation as per ISO 15189, proficiency testing providers test accreditation as per ISO IC 17043, and reference material producer accreditation as per ISO 17034. All the accreditation related queries are available on NABL websites, so where you can access. So far we have accredited more than 7,000 laboratories, but if coming to the Ayush or the drug and pharma laboratories, if I'll see a comparison in for the drug and pharma laboratories, there are around 130 laboratories are accredited for chemical discipline, while in respective with respect to that Ayush testing laboratories are only 48. So there is a very big gap is there, and NABL is working to bring all these kind of the ASU testing laboratories under the accreditation as per the ISO IC 17025. So all these eminent issues like validation, verification, or the stability related issues or the packaging related issues can be resolved, and all the laboratories can come on the single platform with the same kind or harmonization of the methodology. So ultimately, I would like to conclude here. I think I have not much time to speak, but my way forward is that we have a lot still to be done by way of the articulating the challenges that we have until now discouraged the pharma and Ayush laboratories from embracing the benefits of the NABL or the ISO IC 17025 laboratory accreditation. For this awareness and the capacity building with respect to the benefits of the ISO IC 17025 accreditation against the startup cost and other challenges given the concept of testing once and accepted everywhere. So here I would like to request Kasmi Saab to look into that, so we can have the awareness programs for all these testing laboratories of the ASU drugs to aware them to get the accreditation, so that they will come on the same platform with the harmonized methods. And it is a joint call for NABL and also the relevant regulatory boards and the laboratory associations as well. And of course, the PHDCCI is doing a wonderful job to cover these kind of the things under these webinars, so that an improved level of the awareness of the accreditation in the drug and pharma sectors can be done, including the Ayush products. So thank you very much. So here I would like to say at last that accreditation is the synonym of the quality. We can use the accreditation for demanding and recognizing the quality products and to promote the accreditation for ensuring the quality of the ASU drugs. At last, I would like to thanks to PHDCCI, Mr. Patwal, and other eminent personalities to invite the NABL on this very important webinar. And also thanks to Mr. N. Venkateshwaran, CEO NABL, for nominating me to represent the NABL on this webinar. Thank you very much to all.
Thank you so much, sir. Thank you so much for sharing the accreditation process and the importance of the accreditation for the Ayush industry. We can say that quality is the far most important criteria if we want to go global uh, in the Ayush industry. I hope the details and the process you have provided must be very much useful for the Ayush industry member. And also, I would like to urge them to go for the accreditation and the product quality testings and all. And meanwhile, the government will also work upon the uh, laboratory infrastructure development. As you have mentioned, only 48 laboratories are there. Thank you so much, sir, for sharing your insights and the process and details. I would like to now request our chief guest, Dr. M.A. Kazmi, sir. He is the advisor Unani at Ministry of Ayush, Government of India. Request to share your views on the topic, please, sir. Over to you.
Thank you so much. Am I audible?
Yes, sir. Yes, sir.
Okay. Dr. Ravi Rathod ji, Joint Secretary, PhD Chamber. Dr. Raju Rai ji, Chair Ayush Committee and Head Research Tower India Limited. Mr. Pradeep Multani ji, President PhD CCI and Chairman Multani Pharmaceutical Limited. Dr. Pankaj Goyal ji, Deputy Director, National Accreditation Board for Testing and Calibration Laboratory, NABL. Padam Bhushan Vaid Deven Tribuna ji, Patron of the Amam All India Ayurvedic Congress and Ayurveda Mahasammelan. Dr. Santosh Joshi ji, Co-Chair Ayush Committee and Senior General Manager R&D Hamdard Laboratory India. Dr. Devika Gunwan ji, Senior Consultant Ayurveda and Wellness at Max Multi Specialty Center. Mr. J.P. Sinha ji, Vice President Analytical Division, Multani Pharmaceutical Limited. Mr. Shashant Agrawal ji, AGM Quality Control, Hamdard Laboratory Limited. Other dignitaries, ladies and gentlemen, I am very thankful to organizer of this webinar for giving me an opportunity to participate in this prestigious platform. Friends, stability is the capability of a specific formulation in a particular container, closure system to remain within its physical, chemical, microbiological, and toxicological therapeutic specification at a defined storage condition. Stability is expressed in terms of shelf life. Shelf life is the time period during which a drug product is expected to remain within the approved specification for use, provided that it is stored under the condition defined on the container level. Expiration date is the date placed on the container level of a drug product designating the time prior to which the product is expected to remain within the approved shelf life. A specification of stored under the defined condition and after which it must not be used. The concept of shelf life can be traced from classical texts. Similarly, the text of the Unani medicine provides the concept of shelf life. As per Jalinus book Kitabul Murakkabat, the potency of sufuf powder is not more than 2 months and shelf life of bubub and across paste and tablets is more than that of sufuf, whereas the shelf life of the sharbat syrup is about 1 year. Arab physicians Ali ibn Abbas Majusi, in his compilation Kitab al-Mulki, mentions shelf life of across asqil, across afay tiryak arba and tiryak salasa from 2 months to 2 years, and shelf life of majun kibrit from 6 months to 3 years. Friends, as we know that the evaluating the stability of ASU formulations presents a manner of challenges when compared to the chemically defined substances in particular. First, active constituent of ASU formulation consists of complex mixture of constituents, and most cases the constituent responsible for therapeutic effect are unknown. Second, the situation is further complicated when 2 or more herbs are incorporated in ASU formulations. In many cases, where combinations of the herbs are present in ASU formulations, they have similar constituents, and this gives rise to even more analytical challenges. As part of the total quality control strategy of ASU formulation, a set of test criteria, including qualitative and quantitative parameters, has been recognized and quality indicating. With regard to stability test, chromatographic fingerprint as well as appropriate method of assay via marker substances represent the fundamental part of this concept laid down in shelf life specification. Friends, protocol for stability study depends on the various factors like the nature of product itself, the ingredients, the packaging material, and the environmental condition. Stability testing is a complex set of procedure as variety of factors influence the stability of the product. The formulation itself are complex. The degradation reaction may be complicated by possible interaction of several components of the formulation. The Ayurvedic Pharmacopoeia of India, Part 1, Volume 8, under the Appendixes 3.9, prescribed guideline for stability testing and shelf life determination of all licensed ASU medicine. Guideline for stability testing and shelf life determinating of the ASU medicine is first, reaction of the sample and batches. 2 approaches can be followed to monitor the stability of the product. The first approach is to store the samples of the same batch material at standard storage and accelerated storage condition and test them periodically. Based on the evaluation of the result, the expiry date or shelf life may be determined. The second approach, called cross-sectional approach, is applicable for existing products which do not have a declared shelf life. The approach is to select sample from batch to manufactured over a period of last 5 years, spanning 6 months, and evaluate them simultaneously. Second, storage condition. For the stability study, the drug product should be packaged in the same container and closure system as proposed for marketing of doses from which secondary packaging and the container label as appropriate. Each individual strength and the container size of the proposed packaging configuration should be placed on stability unless bracketing, and the matrixing design are used. The change is observed during 6 months for accelerated stability and 1 year for real-time stability. Study at an interval of 0, 1, 3, 6, and 12 months, real-time stability is comparatively carried out to evaluate the actual degradation rate of the product with respect to accelerated condition. Third, testing frequency for long-term studies. Frequency of testing should be sufficient to establish the stability profile of the drug. For drug with the proposed shelf life of at least 12 months, the frequency of testing at long-term storage condition should normally be every 6 months over the first year and the second year, and annually after there through the proposed retest period or shelf life. At the accelerated storage condition, a minimum of 3 time points, including the initial and final time points, for example, 0, 3, and 6 months from 6 month study is recommended. Fourth, container and closure system for the stability studies. The drug product should be packaged in a same container and closure system as proposed for marketing of doses, from which secondary packaging and container level as appropriate. Fifth, specification is established quality criteria to which a product should conform to be considered acceptable for release or use. It is list of test, reference to testing procedures, and proposed acceptance criteria. Stability study should include testing of those attributes of the drug that are susceptible to change during the storage and are likely to influence quality, safety, and or efficacy. The testing should cover as appropriate the physical, chemical, biological, and microbiological attributes. Validated stability indicating analytical procedure should be applied. The purpose of stability is to establish, based on the testing, a minimum of at least 3 batches of the drugs. A reset period applicable to all features, batches for the drug's substance or shelf life, and level storage instruction applicable for all future batches of the drug product manufactured and packed under the similar circumstances. We may follow and stick the testing protocol and stability of ASU drugs so that safety and efficacy of the ASU drugs may be ensured. Thank you and Jai Hind.
Thank you so much, sir. Kazmi sir, for sharing the detailed information on the product shelf life and the importance of packaging and container system, which is very much affected by the lot of environmental factors. We can, sir, as per your views, we can share that the stability test is important, not only in the process of product development, but also to assess the quality of existing products over the period of time. Right, sir? So we can say that when the Ayurvedic products are subjected to stability studies, the dossier of an Ayurvedic product becomes more scientific and acceptable to the customers, also to the industry, or to the patients also. Thank you so much, sir, for sharing your views and the detailed information. We now have with us Dr. Santosh Joshi. He is the co-chair of our Ayush committee and the senior general manager R&D at Hamdard Laboratories, India. I'd like to request Dr. Joshi, Santosh Joshi, sir, to share his views, please. Over to you, sir.
Thanks, Ravi. Am I audible?
Yes, sir.
Yeah, good afternoon to all. Today's chief guest, Honorable Dr. M.A. Kasmi Sahab, Advisor Unani, Ministry of Ayush, Government of India. Guest of honor, Dr. Pankaj Goyal ji, Deputy Director, NABL. Padma Bhushan and Padma Shri, Honorable Ved Devendra Tribhunaji ji, Patron of Amam. Honorable Sri Pradeep Multani ji, President, PhD Chamber. Eminent speakers, Dr. Deepika Gunwan ji, Senior Consultant, Ayurveda and Wellness, Max Multi-Speciality Center. Mr. J.P. Sinha ji, Vice President, Analytical Division, Multani Pharma. Mr. Shashank Agarwal, who is my colleague, ASM Quality, Hamdard Laboratories, India. Dr. Rajiv Rai ji, Chair Ayush Committee. Dr. SPS Bakshi ji and Mr. Jiten Sori ji, Co-Chair Ayush Committee. All leadership team members of PhD Chamber, respected participants. We are thankful to you all for participating in this webinar, and today's topic, testing protocols and stability studies of ASU drugs, is very important for all herbal medicine manufacturers, as stability testing or shelf-life study of herbal products is a mandatory requirement for patent or proprietary products for getting product approval or filing a license for getting the approval of a patent proprietary medicine. In rule number 161B of Drug and Cosmetic Act, there is defined shelf life for classical or Shastri products. But for patent and proprietary products, there is a provision that we have to submit the stability data for getting product approval. Shelf life study is an important component of herbal ingredient for herbal ingredients also evaluation, and equally important for products development process. Drugs regulatory agencies across the globe have guidelines for stability studies of herbal products, and stability data of any product has to be submitted along with the product registration dossier. Shelf-life study of a product is one of the important parameters for ensuring its quality, safety, and efficacy. However, performing such testing on herbal products is not as convenient as with synthetic drug products, where we are having few active pharmaceutical ingredients act as active component, and standardization of that single component is not that tough as we are facing challenges with herbal formulations. Generally, herbal medicines are prepared from multiple ingredients from varied sources. It contains herbal ingredients, minerals, and other components too. Like we are also adding additives and preservatives for ensuring the shelf life, and are sometimes interacting with the actives component and creating some challenges. There are various chemical constituents present in herbal drug which are liable to varied chemical reactions under the influence of different conditions during the shelf life. Such reactions may result in altered chemical composition of herbal drug product, and consequently altered therapeutic profile. So these are the challenges, and the key challenges with herbal medicines are chemical complexity and the variability in biochemical composition of raw materials, enzymatic activity. Initially means during collection of the plant material, immediately after plugging the leaves from the plant, enzymatic reactions starts taking place, and some chemical components changes from their original form to means some transform forms, and selection of markers. These are the key challenges. Selection of test parameters is also very much important as far as herbal medicines are concerned, and has a considerable influence on reliability of data generated for assessment of the shelf life, as lack of identification and availability of therapeutic markers, changing chromatographic fingerprints due to inter and intramolecular interactions among the phytoconstituents, batch to batch variation are some big challenges which we are facing in the herbal products. One more thing I would like to add, during processing also means sometimes some challenges we are facing, like in the classical text some methods are mentioned like Kheer Pak Vidhi, where we are boiling the herbal extract or decoction with the oil. So the resultant products parameter, if we are trying to correlate it with the raw material, and we are trying to standardize our finished product. So sometimes we are facing challenges of correlation of acid value or peroxide value, as already mentioned by Dr. Rajiv Rai ji. So all those challenges we are facing, since therapeutic activity of an herbal medicinal product can't be attributed solely to a particular compound or compounds. That's why monitoring of both chemical parameters as well as assessment of intended biological activity is recommended. to be a part of standard stability testing program of herbal medicinal products, because due to synergistic action, these formulations are giving their therapeutic benefit. We can't say that any single ingredient is responsible for biological activity. So it's very much important that we should also conduct bioassay along with chemical parameters to prove the efficacy of a herbal product. So without taking much time, today we are having some eminent speakers. They are experts in analytical chemistry. They are having very good insight, knowledge about stability data, conducting specific test parameter for herbal medicines. I would conclude, and thanks for allowing me to share my views. Thank you.
Thank you so much, sir, Santosh Joshi, sir, for sharing your views and the key challenges faced by the industry. We can say that phyto formulation is a mixture of more than one ingredient, as you have said. So we should take care to determine the stability profile of the natural medicine, which gives more confidence to the patient. Thank you so much, sir, for sharing your views. Sorry, I was on mute. Thank you so much, Dr. Santosh Joshi, sir, for sharing the key challenges faced by the industry on the stability study tastings and all. You have rightly said the phytoformulation is a mixture of more than one active ingredient. So we should take care on the determination of stability profile of the natural medicine, which ultimately increase the confidence of the modern patient compliance. Thank you so much, sir, for sharing your views with us. Now I would like to request Dr. Deepika Gunawat, ma'am. She is Senior Consultant, Ayurveda and Wellness at Max Multi-Speciality Center. Over to you, ma'am, to please share your views.
Namaskar and good afternoon, everyone. Thank you, Dr. Ravi, for inviting me over. It's always a pleasure to participate in such webinars with PhD. I have a small presentation, Dr. Ravi, and if that could be taken on the screen. It's a very brief presentation. While my learned colleagues and experts have already spoken on a bit on stability and also on the analytical part, I'll just try and take the audiences a little into a little back in time. So can I have the first slide, please?
Yes, ma'am. Is it visible, ma'am?
Yeah, it is. It is. I can see it. I'm sure the participants can.
Yes.
So, why I want to take you back into time is the reason being, why are we talking about all these tests? What's the significance? Why are they mandatory for us now? So I just thought I will briefly touch upon all those aspects. But before I do that and put forward my point of view, I thought we should look at, when it all started. So we all know that the Vaidyas, the Kaviraj and the Hakims had their own setup, and they were controlling the manufacturing and dispensing in earlier times. And even now, few of them would be doing it in certain rural places. And the prescription was always very individual and patient-centric. They used to collect those herbs, dispense it, and give it to the patient. So it was a very small-scale setup. Why I'm saying so is because I have been part of the small-scale setup when I was doing my graduation and post-graduation. And I was fortunate enough to work with two of my Gurujis in Kolkata, Kaviraj Jyoti Prasad Gupta and Professor Ellen Maiti. Both of them have been awarded by RAV in the last few years, I think. So I had really learned the art of manufacturing and how we create those traditional formulations with those traditional concepts. And you will see that the stricter control on RM, the processes and the shelf life of substances was already existing then, because it was very small volumes. I've seen Chyawanprash being done in 5 and 10 kg batches. Avipatti ka Churn 1 kg batch, Haridra Khand 1 kg batch, and so on and so forth. So these products were all made depending upon the patient, their requirement, and how many patients the Veggie or the Kaviraj expected in the next 3 or 6 months. So, and the raw materials were always picked up either by them personally or through an expert in pharmacy and Dravigun. So this was a very small setup, but a very strict control on RM. So this is where it all started. Next slide, please. And it's not only happening, but we also are reading. When we studied that there are 4 important quadrants of therapeutics in Ayurveda. Our book on medicine, Charak Samhita, there is a chapter on Chikitsa Chatushpad, chapter 9, and it talks about physicians, drugs, attendant. Why I'm going to all this is because being a physician of analytical, but I'll be talking about why we want totally deals with the physician, drugs, attendant, and patient. And physician being the top of the pyramid. Then drugs, attendant, and patient. The drugs had to possess these 4 qualities: abundance, suitability, multiple forms, and potency. By potency, we were indicating towards effectiveness and quality. If that did not be there in the drug, then obviously it will not give us the response or give us the results. It will not be effective. Now, this was primarily based on microscopic identification, which was based on morphological features and organoleptic properties. And of course, on the geographical area from where the drug was collected, where it was cultivated, and the Draviguna of Ayurveda, which is Raskun, Virya Vipak, Prabhav, and Karma. So based on that, these formulations were made and effectively giving the response in patients, but at the same time, the quality of the drugs was maintained. Next slide, please. We all know that coming back to the topic, importance of testing protocols and stability, and I have seen that Ayush has done a lot in the last couple of decades, and a lot of tests have been brought forward for various reasons, for the impurities that we see around us, for the environmental toxins that we see around us, for the questions raised on Ayurvedic products. So a lot of things were considered, and testing protocols and stability were considered as part of Ayush portfolio and framework. So all this is already existing, but I think we have to reiterate and say that yes, this has to continue and go on. We'll keep debating on what is right and what is appropriate for ASU drugs. We will keep working on it. It's evolution that's happening. But besides that, I think what is available, we should take use of that, put that to use, and get safe quality products. Now, when I talk about importance of testing protocols and stability, I consider there are 3 important aspects to consider. The first one being, we all know that there are significant changes in environment, air, water, and soil, which has led to contamination of herbal drugs even. So it has not spared the herbal drugs. Only 2 days back, I was hearing this news that life expectancy in India has reduced by 5 years because of pollution. So we know that everything is being affected because of the environmental pollution. Therefore, there is not only the loss of potency, but a source of iatrogenic diseases. So as a physician, we have a responsibility. Iatrogenic diseases are diseases when we give a medication or a therapeutic or a treatment to a patient. Apart from treating the primary disease, we are responsible for creating additional problems and also giving secondary problems. So iatrogenic diseases have to be avoided, and for that we need to have good quality products. The second aspect that we need to consider is that globalization has led to commercialization at previously unimagined scale of magnitude. And we see whole lot of, there's a drastic increase in the volumes that are being manufactured. Now, when you manufacture these volumes, and if you don't have the GMPs, my earlier expert was talking about GMPs and all. I think we need all those. We need GMPs, we need GACCP guidelines, we need GLP guidelines, we need GCP guidelines. These are necessary to keep the principles intact so that we get a good quality, safe product from the commercial sector. So that's very important because globalization has done that to us. The third part is that the herbal medical substances, herbal medicinal substances and herbal medicinal products, they have a very complex nature. And my earlier colleague just mentioned about it. We do understand they are of a complex nature, but that leaving aside the safety and other things, we will have to cover. What is inherent in it, we cannot change it. But to add to the problems, and to do deliberate adulteration, I think that's something that we should not allow. Next slide, please. So to have that quality control of ASU drugs, what do we need to do? I think all this is being already done by the ASU sector. A lot of companies are already doing it. If we do this, we will get safe products, good quality products. So what do? We do proper identification, whether it's microscopic chromatography or chemical test, we should do it. Adulterant testing is a yes, mandatory. Pesticides, heavy metals, aflatoxins, microbial contamination, very important. Stability test, we had a complete presentation on stability. It is necessary to check the quality of herbal medicinal products. We know which varies with time under the influence of environmental factors. And in India, we have varying temperatures, humidity. So under those conditions, the stability of a product does get affected. So we will have to do those stability tests, especially for proprietary products where we are making different combinations and permutations and combinations. While there is also that stability which is for traditional products, which is already defined. I think there also we should be repeating that because obviously of the varying environmental factors in India. Now, these measures not only assure good quality products, but are in no way a sacrifice of the traditional concept, but they are only an assurance. Yes, the product is good, safe, can be used in your patients. And this will also help us in getting into research with products that are of very high quality. And therefore, our objective of research is to validate what has been said by traditional science. So if we want to validate, if the product is not good, we will not be able to get the results that we are expecting out of a clinical research. So for me, quality and safety of herbal products is more important and at the top of the pyramid. Next slide, please. So why test the ASU drugs? Now, there's always a reason will be questioned, why do you want to test the ASU product? So as individuals, we are involved in healthcare delivery. And as a physician, it's a tremendous responsibility to provide safe and good quality drugs to my patients so that they are getting results, so that we have success in treatment. We also want to follow the principle of do no harm. As a physician, my first principle is do no harm. So I was talking about iatrogenic diseases. Therefore, if a product is safe, I will be spared of that. I will not be inducing any harmful diseases in a patient. In Chikitsa Chatushpad that I was just talking about, where we're talking about herbal drugs, there is also mention of physicians. A physician is a very important quadrant in therapeutics. And while they're also talking about physician as a savior of life, prana abhisar, they're also not falling back on saying that there is a condemnation of quacks. Now, quack doesn't mean that somebody who is not qualified. It means somebody who remains ignorant to the science and the recent developments. So we can't stay ignorant and say that, okay, these tests are not required. We don't need stability. We don't need tests to do for the adulterants. I think this is all needed. This is all happening. These are recent developments. While we will keep debating on what is good and what is right and what is appropriate, what is available, I think we must make use of that. Technology is available. Sophisticated labs are available. We will make use of that and see that the ASU drugs are always safe and are of good quality to be used in patients. And our aim is to give success in treatment to our patients. Thank you.
Thank you so much, ma'am, for sharing a brief presentation. And you have rightly said there are some factors like environmental changes, globalization, and the complex nature of the herbal medicine are the main factors which mandate the stability study of the product to have them accepted globally for their safety and efficacy. Thank you so much, ma'am, for sharing a brief presentation and your views with us. Now, I'd like to request our next speaker, Mr. J.P. Sinha. He's the Vice President, Analytical Division at Multani Pharmaceuticals, to share his views. Over to you, sir.
Yeah, good evening, and thank you, Dr. Ravi. First of all, I would like to thank PHDCCI to organize such an important webinar, which is such an important topic, and to invite me to share my views here on this platform. And I would like to extend my welcome to Dr. M.A. Kasmi, Padma Bhushan and Padma Shri Vaidya Devendra Tripunaji, Dr. Pankaj Goyal from NABL, our other eminent speakers, Dr. Deepika and Mr. Shashank Agarwal. I would like to extend my thanks to Mr. Pradeep Multani, President, PHDCCI, Dr. Rajiv Rai, Chair, Ayush Committee, and Dr. Santosh Joshi, Co-Chair. Well, first of all, I'd like to share my views what is the importance of ASU products to be tested and the stability studies thereupon. In recent years, more and more people throughout the world are turning to use medicinal plant products in healthcare system. Worldwide need of alternative medicine has resulted in growth of natural product market and interest in traditional system of medicine. On the other side, testing and quality control activity is increasingly becoming more and more important in enhancing the trust in stakeholders. The variability of the active compound concentrations, microbial infestation, and residual contamination of pesticides, heavy metals, antibiotics, aflatoxins, etc. in herbs or herbal preparations, which are made of natural sources, are due to genetic, cultural, and environmental factors, has made the use of herbal medicines more challenging than it would otherwise have been. Herbal preparations are mainly originated from natural sources in which chemical, biological, and genetic diversity have been found due to geographical, environmental, seasonal, and other variations. These variations may also affect the batch-to-batch consistency of the preparations products. Therefore, testing and quality control has to play and has been playing an important role in streamlining the standardization and eliminating above mentioned variations as well as to batch deviations found in herbal Ayurvedic products. Testing and quality control thus becomes the most important factor in the standardization of these preparations. Proper integration of modern analytical techniques and testing protocol development is an essential part in current scenario. Testing protocols include different types of test parameters which belong to physical, chemical, instrumentation, and microbiological categories. Testing parameters are performed and methods are developed with the help of modern analytical and high-end instruments. I'd like to share my brief presentation also in the form of PPT. So I'm just sharing it. Is this visible to all?
Yes, sir. If you can just make a PPT presentation view.
Can you see it now?
Slideshow. Yes, sir. We can see that. You can make it a slideshow.
Okay, thank you. Yeah, testing protocol for Ayush products. Protocol for testing of Ayurveda, Siddha, and Unani products contain various parameters of testing the different categories, single and compound formulations. Testing protocols of Ayush products were developed by Pharmacopoeia Laboratory for Indian Medicines, Department of Ayush, Ministry of Health and Family Welfare, Government of India, Ghaziabad. The Ayush products have been divided in major categories of single drugs and compound formulations. Formulations have been divided in physical categories of solid, liquid, and semi-solid type. Testing parameters categorized in 5 major types: organoleptic, biological, chemical, physical, and botanical testings. These are the main categories. Obviously, organoleptic evaluations have got the different parameters: color, odor, taste, texture, and fracture. You can see it, color is greenish, grayish, yellowish. It can have any kind of color. There can be a different odor. It should be pleasant. It can be unpleasant. Then there is taste. It could be sweet, sour, bitter, or pungent. There are different textures available in different products. The composition of the products can be fractured also. Botanical type, shape, surface characteristics, transverse section, powder microscopy, stomatical index, Palisade ratio, when inlet number, when termination number, lycosporin spore method for starch gains. These are the different microscopical parameters which we. Oh,
Your
just a minute,
slide
please.
is not moving, sir.
Okay. Okay. Thank you. So these are the different microscopical parameters which we do in the different products. Transverse section of tulsi leaf by microscope, outer microscopy of cinnamon bark, stomatal index by microscopic count method, Palisade ratio by microscopic count method, vein inlet number by microscopic count method, vein termination number by microscope count method.
Sorry to interrupt you, sir, but the slide is not moving, sir.
Now it's moving now.
No, sir.
It's not moving.
No, sir. We are on the second page. I mean, the first page of the presentation, testing protocol for Ayush products.
Just a minute.
So you can close it and reshare it again.
इस स्लाइड मूव नहीं कर रहा है अपने आप, मैं कर रहा हूं तो हो रहा है, बट हम लोगों को नहीं विजिबल हो रहा नहीं। मतलब क्या है, वहां पे जब ये स्लाइड में मूव कर रहा हूं तो उनको दिखना चाहिए। नेक्स्ट रीशेयर करना पड़ेगा। हाँ, रीशेयर करना पड़ेगा। ओके,
It has been moved now, sir.
it is moving now.
Yes, it's on chemical parameters. Slide number 9. Yes.
It's moving.
Yes.
Thank you so much. That's very good. Now we are coming over to physical parameters. There are different physical parameters like moisture content, ash values, extractive values, refractive index, viscosity, flow properties and particle size, and foreign matter. I think it's moving now. Yeah, thank you. Moisture content we do by oven drying method, ash values by furnace ashing method, extractive values by gravimetric process, refractive index by refractometer, optical rotation by polarimeter, viscosity by rotational viscometer, powder flow by Carr's index method, and particle size by sieve method. Now we are coming over to chemical parameters. There are phytochemical parameters in which there are chemical, titration, gravimetry, and spectroscopy methods are used for phytochemical analysis. Chromatographic, TLC, SPTLC, gas chromatography, SPLC, and mass spectroscopy methods are used for biomarker identification. Heavy metals and mineral content we do by atomic absorption spectroscopy, ICP-OES, and ICP-MS analysis method for heavy metal and mineral content quantification. Similarly, pesticide residue we do by gas chromatography mass and liquid chromatography mass methods for quantification of organochloro, organophosphorus, and pyrethroids pesticides, besides other pesticides also. For aflatoxin, we use SPTLC, SPLC, FLT, and LCMS methods for residual quantification of B1, B2, G1, and G2 aflatoxins. For
your slide is again not moving.
It has been again stuck, sir.
It's moving now.
Yes, it's on chemical parameters now.
Okay, thank you. Thank you. कोई बात नहीं, right. Sorry for the interruption. Quantification of quality parameters like acid value, saponification value, reducing and total sugar content, peroxide value, etc. quantification of polyphenol, tannins, sugar, and UV visible scanning by UV visible spectrophotometer, fingerprint screening by quantitative analysis of biomarkers by HPTLC, quantitative analysis of biomarkers by using SPLC technique, analysis of heavy metals, mineral composition by using AAS technique, analysis of trace level of heavy metals, minerals, and other metal components by using ICP-OES and ICP-MS techniques, analysis of pesticides, aflatoxins, and other natural contaminants by using LCMS technique, analysis of radioactive contamination by using scintillation detectors, analysis of alcohol content, pesticides, and residual solvents by using GC-MAS and GC-HS-FID techniques. Now we are coming over to biological parameters. In biological parameters, we are having microbial limit test, pathogen detection, pharmacological screening, and toxicological screenings. In microbial test, we do total aerobic bacterial count or total yeast and mold count. In pathogen detection, we do E. coli, S. aureus, Pseudomonas aeruginosa, and Salmonella. Biological, we also do pharmacological screening in anti-inflammatory activity, anti-diabetic activity, etc. For toxicological screening, we do acute toxicity, lethal dose calculation, severe toxicity, histopathology of body organs. Now, enumeration of total bacterial and total fungal count by using microbial limit test techniques, detection of pathogens in Ayush drugs by using selective media growth and biochemical test methods, in vivo and in vitro methods for evaluation of pharmacological effects, acute and chronic toxicity evaluation by using in vivo animal models. Now, these are the parameters, recommended test parameters for Ayush formulations. We can see the parameters of testing in different Ayush products. In the first column, there are different parameters. In the first row, it is different products and which parameters are suitable for testing in which product is clearly visible in it. These are different slides for different products and different parameters suitable for those products. You can share it later on with the interested people. Now we are coming over to a stability study of Ayush products. The purpose of a stability study is to establish a shelf life and level storage instructions applicable for all future batches of the drug product manufactured and packed under similar conditions. The shelf life thus a manufacturer wishes to declare a shelf life longer than one specified by the notification substituted by GSR 789E dated 12/8/2016 with effect from the event date. What is stability? Now, it's a question. Now, the definition wise, it's the ability of pharmaceutical or Ayush products to retain its physical, chemical, biological, and microbiological properties within specified limits throughout its shelf life. Or, in other words, the ability of pharmaceutical Ayush products to retain its quality, safety, and efficacy throughout its shelf life. Now, there are two types of stability studies we normally perform. The first one is accelerated stability studies, and another one is real-time stability study. A study designed to increase the rate of chemical degradation and physical change of a drug by using exaggerated storage conditions. And in real-time stability study, we do the experiments on the physical, chemical, biological, and microbiological characteristics of a drug during and beyond the expected shelf life and storage conditions, periods of samples under the storage conditions expected in the intended market. Now, Ayush guidelines for stability studies, Ayurvedic Pharmacopoeia of India, Part 1, Volume 9, 2016. As per that, the objective of this guideline is to specify the method of arriving at shelf-life by stability testing. The shelf-life determined by the process mentioned in this guideline can be used to decide the expiry date in case a manufacturer wishes to assign a shelf-life longer than one specified by the notification. GSR 789E dated 12/8/2016. Now the guideline can be used for all patented and proprietary Ayurvedic medicines, both new and existing products. General information on stability, selection of batches, container and closure system, specification, testing frequency, storage condition, and evaluation. Stability study shall be done on 3 consecutive batches as per the API. Each new drug product and or if any changes in. If there is any changes in vendor of active ingredient, formulation of the product, manufacturing procedure of the product, primary packaging material of the product. If there is any kind of changes in this supply, we must perform the stability studies. In addition to this, the first batch of every product should be yearly taken for an ongoing stability studies program. Now the factors affecting stability. The first one is environmental factors, ambient temperature, humidity, and light. Now the product related factors. It's the physical and chemical properties of API and excipients, dosage form and its composition, manufacturing process, the nature of containers closing system, overall migration and leachability properties of primary packing material. Storage conditions. There are 4 climatic zones, 1 to 4, and we belong to zone 4, and we will discuss here the same thing. Hot and humid conditions. Recommended storage conditions in this zone. The first one, accelerated 40-degree temperature plus minus 2 degree and 75% humidity, RH plus minus 5%. And for this minimum time period has been assigned 6 months. Similarly, real-time stability study, there is a assigned condition of 30-degree temperature plus minus 2 degree and 65-degree RH plus minus 5%. And the minimum time duration has been assigned 12 months. Now, selection of product batches. First one is stability studies are conducted on at least 3 primary batches. Second, for new products, pilot batches are used, which are at least 1/10 of the commercial batch size. 3, possibly batches of drug products should be manufactured by using different batches of drug substance. 4th, sample size or quantity to be selected in a way which will be sufficient to test in duplicate for each time withdrawal. Requirements for stability studies. First one is stability studies protocol. Second one is stability studies planner. Third one, stability studies chambers, calibration and mapping records. Stability study chambers, temperature and humidity monitoring records. Daily monitoring, temperature and RH monitoring twice a day, or there should be a provision of online temperature and RH system. Temperature, thermal mapping and RH calibration once a day. Testing frequency for accelerated studies, the testing frequency has been assigned at the initial level after 3 months and after 6 months. For real-time studies, it is at the initial stage after 6 months, after 12 months, and then once in a day afterwards. Now, what are the tests required for stability studies to be carried out for accelerated stability studies in solid dosage form? Solid dosage form, the tablets, capsules, and powder. It's clearly visible here. What are the parameters we have to perform in initial 3 months and 6 months? All the parameters have to be performed on all the occasions, but these are the parameters suitable for the solid form doses. Now, these are the tests required for stability studies. For accelerated stability studies in liquid and semi-solids. This is initial, after 3 months, and after 6 months. There are certain changes in the parameters for liquid and semi-solids. In Grita and Tela, these are the parameters suitable for accelerated stability studies. These parameters have to be performed in initial 3 months and 6 months. For real-time stability study in solid dosage form, again tablet, capsule, and powder. The parameters remain the same. Only thing is the duration is changed. After initial, we have to perform after 6 months, 12 months, 18 months, 24 months, or as per the requirement. There can be a specific requirement also from the different clients. And this is the real-time stability study in liquid and semi-solids. These parameters have to be performed on certain intervals. And these are the parameters applicable for Grita and tail samples, IL and Grita. Now, whatever results come out after we perform a stability study on the products, what is the significance? An Ayurvedic drug can be considered to be stable if no significant change occurs at any time during testing at accelerated storage conditions or at real-time storage conditions. Now, what is significant change? This is the most important part and most important slide in that. A plus minus 20% change from the initial assay value if the drug is analyzed for its marker. Second one, if there is a plus minus 15% change from the initial assay value if the drug is analyzed for its active compound. Third one, appearance of new spots in identification by thin layer chromatography when compared with the sample stored in less than 10 degrees. Variation in the physicochemical parameters, moisture, ash, particle size beyond 25% of the initial value. Failure to meet the acceptance criteria as per individual monographs or specification. These are the important factors. If they the value after stability studies vary according to this, then we cannot say that the product is a stable. Now, importance of stability studies, introduction of new drugs to the healthcare market. This will help in deciding these things. Stability testing ensures compliance with international regulations that form part of the registration process for a new drug substance or drug product, determining the safety and integrity of the drug product and also its shelf life and storage conditions. Ensure validation of product manufacturing process, ingredients used, packaging materials, and desired shelf life. Rightly, it has been mentioned by Dr. Rajiv that packaging material plays an important part because there might be overall migration and leachability involved in the primary packaging material. So it may affect the stability of the product inside it. Increase the product acceptance among diverse customers. Now, these are the references we have taken to finalize these PPT and slides. So it can be shared with anyone if they want to have the reference sources. Now, we just want to show the Multani Pharmaceuticals Limited Analytical Division, which is operating a laboratory which is catering to Ayush products, pharmaceutical products, and also food and nutraceutical products. Multani Pharmaceuticals Limited Analytical Division is approved for the testing of Ayurvedic and Unani drugs by the Directorate of Ayurvedic and Unani Services, Dehradun. Approved for the testing of drugs and pharmaceuticals by Drug Controller and Licensing Authority, Directorate of Health Services, Dehradun. It's an NABL accredited laboratory as per ISO IEC 17025 2017 version for chemical and biological fields. Our scope covers the testing of drugs and pharmaceuticals as per IP, BP, USP, cosmetics and essential oils, homeopathic drugs, Ayurvedic, Unani products, and food and agricultural products. The salient features of our laboratories are: it is designed according to the current EHS norms. Chemical, instrumentation, and microbiological sections are well separated to avoid any cross contaminations. Microbiology section well designed with 3 AHUs and other essential equipments. Team of well qualified professionals, trained internal auditors as per ISO IEC 17025 2017 version, equipped with advanced techniques, instruments according to current regulatory and statutory requirements. Well-defined system in place to handle conflict of interest. GLP compliant, certified by the Office of Drug Controller, Uttarakhand. We use proper CRMs, RSS, impurities, WSS, intermediates to ensure quality results. Our vision, the vision of the Multani Pharmaceuticals Limited Analytical Division is to support in analytical research to government and private sectors in the most viable way, support to academia, research projects, training programs for students and research scholars for proper quality control in ASU, pharma, cosmetics, and food industries. Dedicated support to Ayurvedic and Unani and pharmaceutical drugs promotion programs across the borders by developing the analytical methods, their validations, stability studies, and by controlling the contaminants. Provide training to needy personnel on a very nominal fee with an aim to enhance their skill. Now, the important modern analytical instruments we are having is first one, AAS, atomic absorption spectrophotometer with flame, GTA and VGA, high performance liquid chromatography. We are having 11 numbers with RI, FLD, UV visible and PTA detectors. Fourier transform infrared spectrophotometer, FTIR with ATR, UV visible spectrophotometer, gas chromatography. We are having 4 numbers with HS and auto liquid injectors. We are having HPTLC applicator with photo documentation, double door autoclave, vertical autoclave, BOD incubators, closed system for stability testing, Carl Fisher potentiometric resolution tester, 4 numbers. We are having 2 number of laminar airflow biosafety cabinet, liquid optical particle counter, LOPC, stability chambers, accelerated and real time, 3 numbers with different conditions of temperature and humidity. We are also coming up with new working chamber stability room dedicated for the stability studies of ASU products. We have also come up with a dedicated food and nutraceutical laboratories with high-end CFR Part 11 compliant and modern analytical instruments like LCMSMS, GCMSMS, ICPMS, UHPLC, GCHS, and RT-PCR, and many more to support the testing of majorly food, nutraceuticals, and other products. A complete facility in 9,000 square feet area to provide the testing of food and agri, Ayush, animal feed, water and packaging materials. The laboratory is about to go for some national and international credentials such as ISO 17025-2017, that is NABL in our country, FSSAI, EIC, APETA, EIS, USFDA, etc. in due course of time to cater to the different needs of our food industry, ASU industries, and other nutraceutical industries which are operating in current Indian market. Thank you so much, Dr. Ravi, to provide. Again, once again, I am extending my thanks to PHDCCI to invite me and giving me an opportunity to share my views to all of you. Thank you once again.
Thank you, Mr. Sinha, for sharing a very detailed presentation and very informative presentation with us. Now, I'd like to request Mr. Shashank Agarwal. He is the AGM Quality Control at Hamdard Laboratories to share his views, please. Mr. Sinha, I'll request you to unshare your presentation so that we can hear Mr. Shashank Agarwal, sir.
So, am I audible?
Yes, sir, you are audible.
Yeah, okay.
Thank you, sir.
Is that okay now?
Yes, sir, thank you.
Thank you.
Over to you, Shashank Agarwal, sir.
Okay. My presentation is visible to you. Okay. Good evening, sir, madam, and all the attendees of this webinar. I would like to say thanks to PHDCCI for giving me this opportunity to present here. So today I am going to represent the topic significance of testing protocol in ASU drugs. I hope we all are familiar with this protocol for the testing of Ayurvedic, Siddha and Unani medicine. So now I would like to explain brief about the history of testing protocol. Nowadays, with the growing awareness of the healthcare and safety, most of the peoples are moving towards herbal products. And in last few decades, nowadays with the growing awareness and healthcare and safety aspects, most of the peoples are moving towards the herbal products. In the last few decades, market of herbal and traditional medicines have grown up rapidly. As we all know, China is a major producer and exporter of herbal and traditional medicine in the world, and India has a very little share in this field. India can have a big role to play in this field as Indian system of medicine, Ayurveda and Siddha are 1,000-year-old and time-tested. Similarly, Unani is also an ancient system of medicine migrated from Iran, Persia and Gulf countries to India. Indian herbal and traditional medicine industries have annual turnover of about 4,000 to 6,000 crore market and have a big export potential. And for export to Western countries, for example, America and Canada, their strict quality parameters and quality controls with the safety and efficacy are required. And on this front, our industries are not able to produce the documentary support. Then the government of India and export dealing agency, Pharmaceuticals, are very much concerned to promote the export. And for this, Export Inspection Council is also doing effort to lay down limits for safety parameters. It is also felt that protocol for testing of Ayurveda, Siddha and Unani products are not available, and without which industry and various government laboratories are not able to concentrate more on this issue. Therefore, the Department of Ayush assigned Pharmaceutical Laboratory for Indian Medicine, PLIM, Ghaziabad to develop the protocol for testing. And then PLIM Ghaziabad prepared the draft document, which is then sent to various experts of the field. And after consideration of the comment received from these experts, the final document has been prepared. The present volume of the protocol for testing of Ayurveda, Siddha and Unani products contain the various parameters for testing for different categories of single and compound formulations, limits of heavy metals, microbial load, pesticide residue, and aflatoxins method for determination of these safety and quality parameters.
Sir, sorry to interrupt you. Your slide is not moving, sir. We are on the third slide. Yeah, it's fourth now.
So these methods have been taken from the different official pharmacopoeia like Ayurvedic Pharmacopoeia of India, Indian Pharmacopoeia, British Pharmacopoeia, United States Pharmacopoeia, European Pharmacopoeia, and Chinese Pharmacopoeia. And these methods are well proved and standardized. Now it is moving?
Yes, sir. Now it has been moving.
So as well as significance of testing protocol in ASU drug is concerned, so we must know what is the protocol for testing of ASU, right? So the testing of the protocol of testing of ASU is a written plan which is stating how testing will be conducted, and it includes test parameter, testing procedures, product characteristics, critical quality attributes, and decision points on what constitute acceptable test results. It is also a roadmap or training tool for testing of ASU medicine, which gives you the answer of what, where, who, when, and how. What means the objective of the testing protocol? Where means the scope of that particular protocol? Who means responsibility? Who is responsible to execute the particular task? When it's a time and frequency of the sampling point, and how is the procedure? So procedure will define whether how you can analyze your sample. So all the parameters of what, where, who, when, and now is how is incorporated in the existing protocol for testing of ASU. Again, with this protocol, you may know how from protocol. Once you know how from the protocol, then you can perform the gap analysis and implement the correctives and preventive the reoccurrence of the incidents and meet the compliance in line with the regulations. Next is contents of the testing protocol in ASU. If you will see the content of the protocol in ASU, the first chapter is analytical specification of different formulations. It contains the different parameters of individual formulations. For example, like ARC, Asaf and Arista, churn, plant extract, tail, bhasma, sarbat of syrup, pisti, guglu, lepa, and etc. So here you can understand whether means what are the parameters, critical parameters you have to monitor or test during the analysis. Means for example, in case of Asaf and Arista, there are certain set of parameters, whereas in Churna there is a different set of parameters. So in analytical specifications, you may get the parameters which you have to monitor. In the appendix 1 sections, there is a small brief description of apparatus for the test and assay. So what are the apparatus you will need during the testing? It is defined in this appendix 1. In appendix 2, the testing procedure of the crude drugs, for example, the botanical raw material, like microscopic analysis, macroscopic analysis, determination of foreign matters, total S, acid insoluble S, water soluble S, sulfated S, water and alcohol soluble extractive values, loss on drying, TLC, limit test for microbial load and heavy metals, pesticide residue, aflatoxins, etc. So these means in appendix 2, you will get the testing procedure for crude drugs. Whereas in appendix 3, you will get the testing procedure for the bulk and finished products. For example, the refractive index, weight per ml, specific gravity, pH value, boiling and melting range, optical rotation, disintegration test, uniformity of weight, viscosity, saponification value, iodine value, acid value, peroxide values, etc. And last but not the least, in appendix 4, the reagent and chemicals which you need during your analysis is explained in detail. As I had already earlier shared that these testing methods have been taken from the different official pharmacopoeia like API, IP, BP, USP, EP, and Chinese pharmacopoeia. So these methods are well proven and hence universally accepted. Second thing is this protocol is also helpful to meet the regulatory requirement for the like Schedule T and the national and international regulations. It also helps to identify the adulterant or substitute. The existing protocol of testing protocols in ASU drugs also helps to get the accurate and precise results. For example, people from different industry may have different working style, which may impact on the result. Hence, to overcome person to person and process to process variance, a well-defined written protocol helps to get the accurate and precise result, irrespective of the individual performance. The testing protocol is helpful to provide the training for the correct testing procedure to ensure the testing consistency with the accurate and reproducible result. It also helps to maintain discipline at workplace, no matter who done the exercise or the test matter, but the test method will remain same. It will also help to reduce the wastage helps to boost the knowledge and develop analytical skills. It also helps to build the trust because you are following the standard method and pharmacopoeias and protocols. So whatever the result you are getting, that is authentic. So you are also, that is also helpful for building the trust, which again helps to you to prevent the recalls, unnecessary recalls, and no warnings and notice of concern, no import alert, no non-compliance. So once you will get no recalls, no warning, no notice concern, no import alert, no compliance, so then definitely all these things will impact your business growth. And for employer point of view, business growth is very important. Again, an employee point of view, job opportunity. If you have sound knowledge of the testing methods and analytical techniques, definitely you will get the maximum opportunity. But in last and not the least, I would like to request each one of the chemists who attend this webinar, and please follow this protocol and get benefited because it will help to increase your knowledge and develop analytical skills. Thank you from my side. Thank you very much. Thank you all.
Thank you so much, Shashank Agarwal, sir, for sharing the importance of these testing protocols in ASU drugs. You have rightly said, by performing these protocols, it reduces the wastage and recalls of the products, which will ultimately helps in the final cost of doing business, we can say. Right now, as the industry, the government is more focusing on ease of doing business and cost of doing business. So we can say that ultimately it helps in cost of doing business also. Thank you so much, sir, for sharing your views with us. Now, we are moving, we open the platform for the delegates and our panelists to have any questions for a Q&A session. Sir, we have one question in the chat box. I'd like to request the panelists to answer if they can. The question is, FSSAI formulations also come with Ayurvedic combinations. Does these ingredients, when used together, remain stable or gets deteriorated? I'll request our chief guest if you may please throw some light on that, sir. Dr. Rajiv Rai, sir, if you can throw some light on this.
Yeah, sure. So every organic material in the universe has a tendency to degrade, and it is by virtue of temperature, humidity, and UV light. So even if it is going to be consumed, if herbal material is going to be consumed in the nutraceutical or food, certainly some degradation with due course of time and with the effect of sunlight, temperature, and humidity will be there. So we have to evaluate, but for sure there will be degradation with due course of time. So this is the answer for this question.
Thank you so much, sir. So we don't have, I'll see there. Okay. Okay, so we don't have any more questions in the chat box or the Q&A session. So if our speakers have some question to connect with the chief guest or guest of honor, they can, else we will ask our chair to share his concluding remarks and vote of thanks.
So if no further comment here, I'm just going to conclude it and will formally convey my thanks to all the panelists, especially Dr. Kasmi, Dr. Deepika, Mr. Shashank, Pankaj Goyal ji, Sinha ji, and Joshi ji. And I will like to convey my special thanks for all the efforts which these PHDCCI members like Amit Patwal and Ravi Rathod is making for conducting these kind of informative seminars. It is a very relevant and informative webinar here, and I hope that the participants have got a lot of knowledge, especially with the presentation of Sinha ji, where he has captured a lot of parameters which are very much practically important for the testing of the formulations and their relevance. And even the Agarwal ji has also narrated the details about the testing protocol methodology and the book published by the PLIM in the supervision of Dr. Lohar. But my advice will be here that better follow more on the API part and the appendix part of API, because the details and the requirements and the method mentioned in the API is going to be the mandatory requirement for any of the regulatory requirements. So this book is certainly for the help, but it is not a mandatory book and a mandatory requirement. And this topic is very much relevant. And by following the stability study and the testing protocol methods, anybody can improve the quality of his product, and certainly he can be more able to generate more quality in the product also. So a multi-dimensional quality improvement will be there in the operation, in the raw material quality, in the product quality, and he will be able to continuously monitor the degradation and various other factors which are having the health concern, like Dr. Deepika was narrating that iatrogenic problems should not be there. So if the manufacturer and the quality department is able to monitor that the pesticide contamination, microbial contamination, and the aflatoxin contamination are within the control, are within the permissible limit, certainly these kind of problem will not be there. So Ravi, I will like to suggest that we can share few of the slide of Mr. JP Sinha if these are required by any of the participants. These slides are going to be much helpful for especially for the students if they are attending this seminar, and for the lower scale of manufacturer also. So they can very easily get the idea about the parameters which are going to be selected for different kind of dosage forms. And from all these aspects, I would again like to thank to all the panelists and the chief guest of today's webinar, Dr. Kasmi, and various other panelists. And thank you to the participants also. But one I would like to suggest here, and will request participants to generate more questions here, so that if they are going to generate more questions and queries, certainly their knowledge will be more. By having the queries and questions, they are going to put pressure on the panelists, and certainly they will gain more. So in future, I would like to have more questions and queries from their side. If they are going to have this, certainly we will be able to consider that they are getting the knowledge, and that they are generating the queries also. So thank you again, and thank you.
Yes, sir, it is necessary to make the session more interactive with the delegates and the speakers. Thank you so much, sir. Thank you so much all for joining in.
Thank you. And
thank you, sir.
Thank you so much.
दान करना तो फायदा तो ऐसा है।